So we're going to start with a very quick Genomics 101. I know we have distinguished genomicists in the audience, but just to make sure that we're all on the same page. So when we talk about the genome, we're talking about our DNA. There's a copy of it in every cell in our body. 3.2 billion base pairs of letters in this wonderful double helix arrangement. And what we're doing is looking at tiny differences, tiny glitches in that DNA to try and understand what's going on with a given patient in a given context, or to try and understand at the level of science or new diagnostic development or new therapeutic development how those changes in the DNA play out in other ways through our body. So to bring that to life with a real story, this is Jessica on the left, who was 4 years old when she crossed paths with Genomics England, suffering from those symptoms that you can see, a series of developmental challenges. And we sequenced Jessica's genome. People talk about, oh, you know, we share 99% of our DNA with a banana, or, you know, all of 99.5% of everyone's DNA is the same, and so on. When there are 3.2 billion letters or base pairs in the genome, even those tiny percentages add up to huge amounts of difference. So 6.5 million variants in Jessica's genome from the sort of reference genome. One of the first questions we ask is, how unusual are those? So, you know, 2/3 of a million were rare. Okay, we're still hunting. Of those, we can look up in all these different knowledge bases and say, okay, just over 2,500 of those are linked to some kind of change in a protein, you know, DNA to RNA to protein. Which ones of those are de novo? So we not only sequence Jessica, but we sequence both of her parents so that we can understand which ones are novel for her. And then one was a gene that was linked in our own knowledge base to link to her specific condition, which is the deletion of the SLC2A1 gene. And in this case, the treatment was both very effective and also non-pharmaceutical, change in her diet, which is fantastic. So when we're talking about DNA, we're talking about using DNA to try and change these kinds of human stories. And in fact, the origin of Genomics England starts with a human story that Nicola is going to tell us.
So Genomics England began with the 100,000 Genomes Project, and it actually started with Prime Minister David Cameron right back in the 2012 London Olympics. He brought together scientists and clinicians from around the world and said to them, you know, what is the next big thing that we should be doing? And they told him that now was the time for a transformation in bringing genomics into healthcare. The technology was starting to be there, but also the UK was the right place to be there to do this, because we not only had some of the scientific capabilities, but we also had the NHS, a single integrated healthcare system. And this captured his imagination because he had a personal experience of how devastating living with rare disease could be. His son had a very rare form of epilepsy and sadly died while he was in office. And so he did something visionary, and he committed to the UK sequencing 100,000 rare disease and cancer NHS patients. And this was a phenomenal population health study. It involved 97,000 patients and family members, 13 genomic medicine centres which were set up across 98 NHS trusts involved in recruiting thousands of NHS staff and researchers, and enormous amounts of data. And this has led to already some extraordinary outcomes from that study. Recruitment finished in December 2019. If you can go on to the next slide, please, there, Chris. And we're still looking at what the outcomes have been, but a landmark study that was published in the New England Journal of Medicine found that 25% of rare disease patients had a diagnosis for the first time. 14% found variants in regions that wouldn't have been found by other diagnostic methods, and that there really is a clinical reason to be using whole genome studies in clinical care. You can go on to the next slide, please. But the reason for setting up this 100,000 genome study was not just for research, as impressive as that was. It was to integrate healthcare, integrate genomics into healthcare. And so Chris is now going to talk to you a little bit about how we integrated that study into a platform that could deliver for patients.
Sure. And so I've been told before that this slide is a bit hypnotic. So if anyone sort of finds themselves, having strange feelings, then that might be why. As Nicola says, the 100,000 Genomes Project was in effect a massive proof of concept study where the concept we were trying to prove was, is whole genome sequencing useful in the clinic? And so we continue to try and bring together the worlds of research and the world of healthcare, as this diagram kind of shows. So if we think about the work in healthcare, we start with a patient who maybe has a series of challenges, symptoms like Jessica's, sees a doctor, take some blood, sequence it, generate all the data. We then run that through our bioinformatics pipelines. We run it through genome analysis routines, and we find the results. We publish those back to the NHS. They validate those and pass them back to the doctor, who can then treat the patient in the best way. And that kind of closes the loop on the healthcare side. We're currently doing about 5,000 patients and families a month through that, just to give some sense of scale. And it wasn't long ago, for those who are involved in genomics, that doing 5,000 genomes at all ever was a big deal. That's now kind of the monthly flow through that service. We can then de-identify all of that data and make it available to researchers working on either fundamental science, novel diagnostics, novel therapeutics, and critically, we can pass the insights from those research efforts back into live healthcare, whether that's at the level of an individual or at the level of a new treatment or a new approach. If we think about what that looks like across the whole of the UK ecosystem, we're working with all of these kinds of partners and many more. And I loved the talk earlier about systems thinking, because the way that we're trying to transform lives and healthcare in the UK is very much a systems challenge. It's not something that we at Genomics England can do in ourselves. We have this very close partnership with the NHS and everyone else that you can see on this page. So it really is a team sport. When we look at how this plays out in healthcare now, the NHS has in some form or another been doing genetics since its inception a long time ago, looking at sort of chromosomes under microscopes and so on, all the way through to gene panels, SNP tests. We're now doing whole genome sequencing in live clinical care. Large panels for cancer and so on. But the bits in green here, we're already sort of piloting, exploring the clinical validity of, and trying to bring into the clinic. So there's this constant flow of science turning into innovation, turning into engineering, turning into kind of clinical care that we try to keep kind of moving forwards. But I think as well as the science, I've put this slide in just to make the point about healthcare transformation is also about really mundane things, like you have 250 large hospitals in the UK, just in England. And we're trying to, for instance, create new pathways for cancer treatment where the tissue gets frozen so that we can get a better quality genome out of it. And then you have to try and figure out, okay, where does the freezer go in this hospital that's already full of stuff? And so a lot of the labs that we work with still have things on the door like photocopier room. And actually a lot of it is just really about sort of the shoe leather of changing the way that these healthcare systems operate on the ground at national scale. The way that we're seeding those changes, though, is very much coming from the insights that come to us through our research communities, and Nicola will say a bit more about that now.
So Chris has described there in that infinity loop on the left-hand side that one of our founding missions is to create one of the best data environments for research and to create a sustainable clinical return for our participants through diagnostics and therapeutics, a clinical return for the NHS. Every healthcare system is experiencing, huge productivity challenges, and we can reduce the diagnostic odyssey. A rare disease patient has about 67 appointments over 75 months. We can reduce that. We can target cutting down the time to find the right therapeutic for cancer patients. And we want to return for UK PLC, more clinical trials, more R&D investment. But we have made, first and foremost, a commitment to our participants that research will happen in a safe and secure environment. And we've created a research environment on a cloud-based system which happens at a top level. We describe it a little like an aquarium, which is why that icon is there. When you go to an aquarium, it is polite etiquette to look at the fish. You can study the fish. You can make friends with the fish. But it is not polite to reach inside the aquarium, pull out a piranha, and take it home. You will not be invited back. So you can study our data. You can take away the outcomes of your study, but you cannot take our data away. Onto the next slide, please. And so we understand that genomic data is very valuable, but it is only really valuable when joined with other modalities. And so we're working very hard to enrich the quality of our data. And the UK is a really good place to do this with great other national data sets. And in particular, a recent partnership we've formed is with the National Pathology Imaging Cooperative. And they are going to be scanning and digitizing the data from the 100,000 genomes cancer patients. That's about 15,000 patients who have 20 cancers across 84 hospitals. By the end of that, we'll have 250,000 very high definition images. It will be the largest data set of its kind, and joined together with the other work we're doing in transcriptomics, proteomics, and other areas. This is why it is so exciting to be doing research in our data set, and why we have over 600 research projects, over 1,800 researchers from academia and from the commercial environment. And some of the outcomes from this data are extraordinary. Onto the next slide, please. For example, we recently had reported at our research summit in the summer from Dr. Jack Bartlett, who's a consultant pediatric hematologist at GOSH, and he was telling us that running a study on clinical impact of whole genome sequencing on his patients, he discovered that in over 80% of cases, it improved the standard of care. In over 24% of patients, it changed the care pathway, and in 14% of patients, it actually changed the diagnostic. You can also see there a picture of baby Oliver. Baby Oliver was born with an over 6 centimeter tumor on his leg, and the standard pathology and molecular tests were inconclusive, and so normally he would be put onto a pathway for chemo and surgery. However, he had a doctor who was familiar with Genomics England. The whole genome sequencing test found that his tumor was benign, and so he was saved that intervention, which is extraordinary. One of the other things that we do is we have strategic partnerships with major farmers, 8 out of 10 farmers, and one of our pharma partners, through that strategic partnership, has found over 9 novel variants associated with the disease which they're studying. One of those has now passed into wet lab discovery for a specific disease indication. The lead investigator in that study has said that this has completely transformed his understanding of the role of genomics and genetics in drug discovery. But what is particularly high quality about the way in which research can be involved in discovery is that we not only have the data and increasing quality, but we also have some of the lead scientists across the global leading scientists who can work with you to drive that discovery forward, and we have some of those on this slide here.
Thanks, Nicola. And so in the terms of NextMed, the stuff we've just been talking about is basically now, right? Now we're doing 5,000 patients a month through whole genome sequencing in rare disease and cancer. Now they're getting diagnosed and improved on standard of care, as Nicola said. Now we have thousands of researchers accessing these data sets, and now we have genomic data linked to clinical data, linked to imaging data, linked to transcriptomic and proteomic data. All of those things are happening today. When we look ahead, we're really focused on how can we use these kind of insights and the infrastructure that we've built to try and capture some of these concepts around predictive health, personalized health, precision health. And one of the ways that we're doing that is with a major newborn sequencing program. Some of you may be familiar with Robert Green's work in Harvard around BabySeq. This is kind of conceptually similar to that, but on a population scale. So we have the funding to sequence 100,000 newborn babies, and to do that really with 3 aims. The first aim is to pick up any of around 250 conditions at birth that are driven by the genetics, that are treatable, and that affect children under 5. So we're not looking at risk of cancer in later life. We're not looking at risk of cardiovascular disease in later life. We're looking at diseases that hit kids and diseases that are treatable. So if a baby is sequenced, they have one of those 250 conditions, they get straight onto the right treatment pathway in the NHS. So that's kind of aim 1, is screening for those conditions. Aim 2 is, if you're a baby that has a genetically driven disease that affects young children, but for which there is no cure, you're not going to get onto those pathways because there's nothing we can do at the moment, but we want to use this as a real accelerator for therapeutic innovation, particularly for platform-based therapeutic development for kind of small n and n of 1 drugs that we can use to effectively add more conditions to that list that can be treated at birth. And the third aim is the one that's kind of visually represented here on the slide, is this concept of the lifelong relationship with the genome. What does it mean for you as a kid who presents in intensive care to have your genome on file? How can that accelerate getting you the right treatment, the right therapeutic at the right time? Into teenage years, what does that mean in terms of mental health, hereditary diabetes, obesity? Into middle age, cancer, cardiovascular disease. Into later years, neurodegenerative disease. So this will be the first cohort of humans born with their genome on file. And so one of the questions we're going to be exploring over the next basically 100 years is, what does that mean, and how can this relationship with the genome help us all to live the sort of healthiest, wellest, best lives that we can lead? For me, it's particularly useful because when our paymasters at Treasury ask me for the KPIs, they say, come and ask me in 100 years and we'll know whether this has succeeded or not. But joking aside, we think that the potential here to really transform lives at scale and to demonstrate the concept that these technologies can really help people over the course of their lives is truly extraordinary. But at the heart of all of that is sort of back to people. Nicola, maybe just a sort of closing thought on how we think about doing that, because tons of risks involved.
So we obviously operate right at the edge of genomic integration into healthcare, but also innovation with the newborn screening program. And the only reason that we have maintained trust, not only from patients and the public, but also from successive prime ministers and treasuries, is because we have integrated deep into the heart of the program that we run our participants, not just through the design of our programs, but actually into the structure of Genomics England. We have what's called a participants panel. It's 25 patients who were involved in the 100,000 genome program, but also now the ongoing programs, which have been a COVID program and also the Genomics Medicine Service. And through that, they hold the board to account for the way in which we run our system. And it is through that we know that everything that we do is being scrutinized by those who have the most at stake. And that way, we have the greatest advocates at the heart of what we do, and we know that we will deliver for those who need it most.
Thank you very much.
Awesome. So I love how you have patients included. How do you get clinicians included, this whole idea of workflow, to integrate all this into the day-to-day life of a primary care doctor? Any quick example?
Yeah. So very quick example is we have hired a bunch of amazing UX people, user experience people, from places like Google Health and other places. And we've embedded them with the clinical teams that are delivering this care so that we can try and bring these insights as seamlessly as humanly possible into those systems. Back to the examples earlier about fax machines and so on. That's not easy, but it's something that lies at the heart of how we interface with the healthcare system.
We also hire actual clinicians who can talk to other clinicians and demonstrate the value of the program. So when I talked about Dr. Jack Bartlett's study, he can go and advocate and talk about how this has completely transformed the work that he's doing on the ground. And I always think that those who are living the experience are your best advocates.
Absolutely. Well, we'll be back in 100 years for the update. Thank you so much.
Thanks.
All right.